All right, guys, back in the van with you today. Yesterday we left off with the process of transcription, talked about some details surrounding the process of transcription, and also what happens to the mRNA strand after transcription occurs, and the fact that it gets processed relatively extensively to the point where you add a couple of what are known as a guanine cap and an poly A tail, and then you cut out some introns and leave just the exons, so you get something we know to be called just regular mRNA. From that point on, we are going to take that mRNA out of the nucleus. At that point, the mRNA has come out of the nucleus, and we are going to translate it. The process of translation is, as it states in the title of this slide, the RNA-directed synthesis of a polypeptide. So we're going to take that mRNA and get something tangible from it. In other words, we're going to get a protein from it. Now, there are certain very important molecular components that are part of translation, and that's what we're going to talk about it briefly in this slide. A cell translates the mRNA message into protein with the help of tRNA, which I touched on very briefly yesterday, as well as ribosomes. And ribosomes I know you've heard of. We talk about them all the time when we get into cell organelles. But tRNA is mentioned a little bit less than we normally would. Okay, but it is extremely important. So we're going to start off first discussing what tRNA is. The molecules of tRNA are all distinctly different, and the reason that they are distinctly different is because they actually carry specific amino acids. Each tRNA attaches to only one of the 20 known amino acids, so it makes so that each one of them is actually unique. The tRNA molecules have something known as the anticodon. The anticodon you see right here matches up complementary with the mRNA. So as we saw yesterday, the mRNA bases match up complementary to the DNA when the RNA is created. And now we have something else that's complementary, but in this case, it is the tRNA that is complementary to the mRNA, and it is complementary in something known as the anticodon. Okay, so this picture right here gives us two looks at what our book shows as tRNA. This is what it would look like in a three-dimensional structure. It's all coiled up and it looks all knotted and raggedy. But basically what this is it's a stretch of RNA. Okay, we have the 3' end over here, kind of coils all the way around here, makes a little loop and goes back up here, and ultimately ends up here with the 5' end. Okay, now at one end of this, we see what's called the anticodon. Okay, this, if we look at it closely, is a 3 base pair. Okay, it's a 3 base pair of RNA that matches up complementary to one of the codons, one of the 64 codons that our mRNA could have. Okay, and at the top here is where we attach the actual amino acid. Okay, what I like to call these things, I call the tRNAs. I think of them as the UPS drivers, and now it's like the Amazon drivers. I think of them as the Amazon drivers of this entire process. They bring the little package known as the amino acid to the place that it has to be and deliver it to the exact spot that it needs to be at. Now, this one over here, this picture here is just kind of an idealized version of what you see over here. Just kind of gets rid of all the tangles and makes it look real nice. And down here we see the anticodon, and up here we see the amino acid attachment site. So TNA is really important. Accurate translation requires 2 steps, and they both involve tRNA. The first step we have to think about is the correct matching between a very specific tRNA molecule and a very specific amino acid. You have to have the proper tRNA molecule match with the proper amino acid. Otherwise, it doesn't matter what your DNA had in the code or what was coded for in the codons of the mRNA. You're going to get the message wrong. So if you don't actually have proper attachment of the tRNA to the specific amino acid that it should have, you're going to get really, really bad results in the end. Okay, second step is you need the correct match between the tRNA anticodon and the mRNA codon. And so this first picture right here goes with the first step that we saw previously. Let's get back here. Okay, showing the correct matching between the tRNA and the specific codon. We're not going to go through this process or these steps, but it shows you that it entails using an enzyme. Okay, this kind of white half circle thing up here looks like a broken egg, is an enzyme that will take a very specifically shaped tRNA molecule and bind it to a very specifically shaped amino acid. Okay, and so that is how we get the proper attachment. Hold on here. All right, I had to take a phone call. It was Mr. Christopherson, actually. All right, so we talked about the proper attachment. These enzymes are shaped in just the right way, and we know that's how the active sites of enzymes actually work, so that you can actually make sure that you bond the proper amino acid to the proper tRNA molecule. Okay, so that is the proper attachment. And next, once we have the proper attachment of the amino acid to the proper tRNA, you absolutely have to have or make sure that those match up properly with the codon of the mRNA, which is what we see in this picture right here. And this picture right here, we see the mRNA down at the bottom, okay, right here. And we see these little tRNA molecules flying in here, and they will match up complementary to what we see in the codon. So if you can see this codon right here where the pointer's at the bottom, it says from right to left CGG. And if we look at the anticodon, it should be complementary and match up. That's how we know, or the cell knows, that it's bringing in the proper amino acid. So it says GCC, okay, and that matches complementary, and so that tells us that this particular codon, for whatever reason, wants the amino acid glycine to be next in the polypeptide chain. Okay, so now we're going to discuss a little bit more about, don't want to miss this, okay? So this is, again, something we've seen before. This has all of the codons that are coded for in the mRNA bases, and it shows their amino acids that they will code for. Now, for instance, this one here, leucine, that we're looking at right here, this amino acid is actually brought into the ribosome and attached to the protein strand by a tRNA molecule that will match up complementary to one of these 4 codon sequences. Okay, so this amino acid right here is actually brought in by a tRNA molecule that matches up with one of these 4 codons. Okay, now if I can go back a couple of slides, and I probably should have mentioned this before, but it's more pertinent now that I discussed that. That third base in the anticodon can wobble. Okay, it's not like a weird dance or something like that, but it can move around. And what that does, as we talked about redundancy yesterday, in this case, the fact that this can wobble allows for some flexibility when they're matching up with the codons, and so it allows for instance, let's say here, if there are mistakes, you notice that this last base pair in the codon, they're all different, but they code for the same amino acid. That is where the wobble effect comes into play, and that it doesn't really matter what the third one is. Okay, the third one could be anything. If it's there, it's going to get leucine. As long as it's CU and then something, it's going to get leucine. So that wobble effect means that the third base pair doesn't matter all that much, and it provides some flexibility in terms of making sure that you get the right one. Okay. All right. So we also mentioned that the other molecular component that is important for translation is known as the ribosome. And so I'm going to talk real briefly about the ribosome and what that is and the structure of it. But the ribosome does help specific coupling of tRNA anticodons with the mRNA codons. Remember, we talked about how important that match is. If that match doesn't happen, then you don't get the correct amino acid in the growing polypeptide strand. And that's what the ribosome does. The ribosome helps to match those things up. And now the ribosome, most of you guys see the ribosomes in pictures, and I'll show you, that picture right there, for instance, the one I just flipped to. The one looks like a little brown nugget, like a little turd. That's a ribosome. But what a ribosome is actually made up of is RNA. Okay, it's made up of a specific type of RNA called rRNA. Okay, and in this picture here, if you see these kind of blobby structures down here, that's the small unit. It's kind of peach or white in color. That's the small ribosomal subunit. And then this blobby thing up here, this big blue blob, is the large ribosomal subunit. And they're shaped this way, this weird three-dimensional structure like that, because the RNA that makes those subunits up is highly folded and highly blob-like, okay? Highly globular is what they would call that, all right? So the ribosome is actually made up of RNA itself, okay? Most of the pictures we see just don't point that out. They look like these little turd things, but you need to make sure that you understand that these subunits here, the small subunit and the large subunit, are actually made up of highly coiled RNA. All right, now the ribosome internally has a few distinct areas. It has 3 binding sites for tRNA, and this helps with the organization of the polypeptide, the growing protein strand, as well as the organization of things happening inside of the ribosome, okay? Now, we can break these up into 3 different distinct sites, okay? We've got the E site, the one all the way farthest to the left, okay? We've got the P site and then the A site. The E site stands for the exit site, okay? This is going to be the site where, well, I'll explain a little bit longer, or a little bit later, but the amino acids, the tRNAs, that get used up will exit here. They will be kicked out, and so it makes room for a new one. The P site, I don't call it the peptidyl tRNA binding site. It's too much of a mouthful. I say that the P in P site stands for polypeptide or protein site. In the diagrams that you'll see here, you'll notice that the P site holds the growing polypeptide strand. Okay, it's kind of where it's set up, and you see it kind of will come right out in the middle in most of these diagrams. The A site, it stands for amino-acyl tRNA binding site. I call it the amino acid site. A site, amino acid, it's easier to remember, and really it makes more sense as to what it actually does. The A site holds the tRNA that carries the next amino acid that will be added to the growing polypeptide chain. Okay, so you can simplify what these do based on the letters. Okay, the A site brings in a new amino acid. The P site holds the growing polypeptide chain. The E site kicks out or makes the tRNAs that have been used up, makes them exit the system. Okay. So this picture here shows the ribosomes with tRNA bound up into the different sites, just so you can kind of see what I was talking about. We see in the E site here, it has been kicked out. Okay, this used up tRNA has been kicked out, thrown away, no longer needed. The middle site, the P site, holds the growing polypeptide strand we see right in the middle. And then this tRNA here, this is the newest one to come into the system. It has brought a new amino acid. This is the next amino acid that's going to be added to the chain. And it will be connected, this part of it right here where the pointer's at will be connected to this part here. And then the whole thing will shift over, okay? So now translation happens in 3 distinct stages, which we'll talk about here briefly. Number 1 is called initiation. Okay, first thing we have to do, you remember that ribosomes help to translate mRNA, right? At this point, RNA is all by itself, ribosome is all by itself. So the first thing that has to happen is these 2 things have to associate, okay? So the small ribosomal subunit is going to bind with the mRNA and a special initiator tRNA. So you get these 3 new things kind of bound up together. You get tRNA, you get the mRNA, and you get the small ribosomal subunit all bind together in a process known as initiation, okay? Then the small subunit moves along the mRNA until it reaches what's known as the start codon. Every single amino acid in your body starts with the exact, every single polypeptide in your body starts with the exact same amino acid. It always starts with methionine, okay? And methionine is coded for by AUG. This lets your ribosomes and tRNA know that the gene has started. So it makes sense that every gene, every protein should start with it so it can tell when it should start doing its job, okay? This one I'm less worried about here. Proteins called initiation factors bring in the large subunit, so the initiator, blah, blah, blah. What you need to know from that is that the large subunit does ultimately come in and bind up to the smaller subunit, so we get something that looks like this, okay? Now, this is the process of initiation. We have that initiator tRNA. That is the one that carries methionine, okay? Methionine is the amino acid that starts off, again, every polypeptide in our body because it will always attach to this AUG start codon, okay? So the mRNA will bind to the small subunit here. It will actually move through, okay, until it reaches this first start codon, which is right here. At this point, once that start codon has been found and attached to and read, the large ribosomal subunit comes and sits right on top. And we notice that this tRNA right here with we'll call this a protein strand, even though it's one amino acid, sits right in the middle. This is the first amino acid in the polypeptide strand. Okay, next we have elongation of this polypeptide chain, where each addition of an amino acid involves proteins called elongation factors, which we don't worry about those. But elongation occurs in 3 steps of its own. First, you have to recognize the codon. You form a peptide bond after that, meaning you are going to link together 2 amino acids, right? Those bonds, those peptide bonds, are the bonds that link together amino acids. And then we translocate, which means to move the mRNA strand over one codon so that we can actually get a new tRNA in, okay? So let's start up here in this picture here. This is a cycle that you'll have to know for the test. So make sure that you are looking at this and make sure you understand that it is important, okay? We see up here, initiation has happened, okay? We've got a growing, in fact, we've got a lot of elongation that's already occurred. Right in the middle in the P site, we notice that the growing polypeptide strand sits. Okay, that's what all these little purple shapes are right here. All right. We will also notice that this tRNA comes flying in here, the little UPS Amazon truck with its very specific package, is going to fly into the A site, the site for the new amino acids to be delivered. Okay, this is like the doorstep. So the tRNA brings in the amino acid to the A site. It makes sure that it binds up or matches these codons complementary. So these codons right here, or this codon right here, matches up with this anticodon right here. Okay, so we see the tRNA sits in the A site there. Next, we have a transfer. You see here a transfer of this growing polypeptide strand to this amino acid that was just brought in. And so we have a peptide bond formed between this diamond amino acid and this circle amino acid right here. And we see now the entire new peptide strand has been kind of moved onto this tRNA. At this point, we have the process that was in the previous slide called translocation. And what translocation means is that this entire complex that you see right here is going to move over one spot for each one. So this empty, used up tRNA is going to move into the E site, and then we're going to kick it out. There it goes. And then this polypeptide attached to this tRNA moves over to the P site, which is what we see happen right here. Okay. And so the entire process continues over and over and over again until we get the entirety of our amino acid chain linked together. All right, finally we have termination. Termination is not as easy as it was in transcription. Termination occurs when you get a stop codon, okay? And there are 3 different stop codons. I'll go way back here. Oh, there we go. Hey, there are 3 stop codons: UAA, UAG, and UGA. All right. When one of these 3 codons comes up in our mRNA chain or our mRNA strand that is being read by the ribosome, like we have right here, that's when termination actually occurs. Okay, so when the termination occurs, when the stop codon reaches the A site of the ribosome, the A site accepts a protein called a release factor. The release factor causes the addition of a water molecule instead of an amino acid. And what we actually get here is something that we've covered before, we've talked about. When you have this release factor right here, it actually binds a little bit of water here, and what it does is it cleaves this bond right here. And if you remember back again to chapter 5, these bonds that get cut by water, that process is known as I've done screwed it up. Hydrolysis. Splitting by water. Hydrolysis is splitting by water. So the release factor brings in a water molecule. The water molecule cuts this bond right here, and then the polypeptide falls off. Then everything else, once the polypeptide's gone, everything else dissociates and breaks into smaller pieces. Chances are this mRNA molecule is going to be digested by the cell. The nucleotide base is going to be used for something else, and the entire process will then start again. Okay? So termination of this process is a little bit more drawn out than it would be in transcription because we have to release that protein from the entire complex. All right. This picture here shows everything. I think it's a great picture to study from. You go all the way up here in the upper left where you have transcription of the gene. You have even talks about RNA processing. It shows the introns and the exons here. Then we have our modified or processed mRNA leaving the nucleus. We have that attached to the small ribosomal subunit, as we see right here. The large ribosomal subunit comes in, and we get our polypeptide strand that grows. So I think this is a really good diagram to kind of quiz yourself or narrate and see if you actually understand what's happening in each phase. Okay? All right. So tomorrow, I guess that would be Monday, we're going to finish up chapter 17 by discussing mutations. Okay, mutations are actually really quite interesting, and I'll have another couple of assignments to go with this lecture that we have on mutations. All right, so I will see you guys then.